Within a eukaryote cell lies the nucleus, made up of the nuclear envelope, the nucleolus, and chromatin. The nucleus holds all the genetic information in a long molecule of DNA. A gene is a short section of DNA that codes for synthesis of a specific protein. The central dogma states that DNA makes RNA and RNA makes proteins. This occurs in cells through a process called transcription and translation. DNA is a double helix made up of complementary base pairs A, T, and G, C. It has a strand that runs from 5' to 3' and an antiparallel strand that runs from 3' to 5'. Transcription of DNA produces RNA in a three-step process: initiation, elongation, and termination. Initiation occurs in the Tatar box, a specific combination of nucleic base pairs. This is what the enzyme RNA polymerase knows where to begin. RNA polymerase unwinds the DNA double helix and separates the weak hydrogen bonds between nucleotide bases. It also produces RNA, a copy of the DNA. There are some key differences between DNA and RNA. RNA has the sugar ribose, it is single-stranded, and has a uracil instead of a thymine. The elongation phase produces the RNA in an antiparallel strand to that of the template DNA. If the template DNA strand is 5' to 3', the RNA will be 3' to 5'. This RNA strand is called messenger RNA.
When RNA polymerase reaches the termination sequence, the transcription process stops. The mRNA strand is released from the DNA strand, RNA polymerase leaves the strand, and the DNA strand recoils into its double helix shape. The resulting mRNA strand seen here is labeled pre-mRNA, since in eukaryotic cells it must be processed in the nucleus before it can leave to be translated into a protein. To process the mRNA, first a 5' cap and a 3' poly A tail are added to the pre-mRNA strand. These prevent the mRNA from degradation and help export it out of the nucleus. Secondly, the mRNA is spliced. Sections of the strand called introns, which aren't needed for the protein that's being coded for, are taken out by an RNA enzyme called a spliceosome. The segments that remain are called exons. Splicing means that one section of genetic code can produce multiple proteins depending on the exons that are chosen. At this point, the mRNA molecule is ready to leave the nucleus out of a nuclear pore. Once in the cytoplasm, the mRNA binds to a ribosome, which is either attached to the rough endoplasmic reticulum or free in the cytoplasm. Ribosomes are also a type of RNA enzyme which translate mRNA into protein through translation. They are composed of a large subunit which binds to incoming tRNA molecules and cycles them through the A, P, and E sites, and a small subunit which binds to an mRNA strand.
The tRNA molecule has a folded structure with the 3 loops, which is in the shape of 3-leaved clover. Transfer ribonucleic acid decodes a sequence on mRNA into a protein. Basically, it helps in translation, a process which synthesizes a protein of mRNA. Each codon is recognized by specific tRNA. One of the 3 loops contain a sequence called anticodon, which recognizes and decodes mRNA codon. Usually, start codon is AUG. Each tRNA has its amino acid attached at its end.
So now we're going to talk about elongation. The first step of elongation is codon recognition, which happens when the amino acid tRNA binds to the ribosome and is complementary anticodon to the codon of the mRNA in the A site. The second step is peptide bond formation. A new peptide bond is formed between the polypeptide in the P site and the new amino acid in the A site. Later, translocation happens. That is when the ribosome shifts towards the 3' of the mRNA by one codon or 3 nucleotides length. The tRNA that was in the A site is now in the P site, and the tRNA that was originally on the P site is now on the E site, which stands for exit. The A site is now empty and ready to bind to a new amino acid tRNA. This process happens until the stop codon is reached. After elongation, we have termination, which is when a stop codon is detected and a release factor RF binds to the codon. We can have a closer look down here. The stop codon can be either UAG, UAA, or UGA. RF promotes the hydrolysis of the bonds between the tRNA in the P site and amino acid, freeing the polypeptide from the ribosome. Later, all components dissociate. After translation, many proteins require additional modifications before they become mature proteins, such as addition of sugars, lipids, and phosphate groups. There can also be structural modifications like cleaving of parts of the peptide and bringing multiple polypeptides together to form a quaternary structure.